glyphosate substitutes for glycine, and if that's right, the implications are not just in one disease or one part of the body. It's everywhere, and there's not much you can really do about it.
Right. Yeah.
Can you talk a little bit more about how it's substituting for glycine?
Yes, this is the thing that, Anthony Samsel and I collaborated on a number of papers, and he suggested this to me in December, I think, of 2015. I had been kind of toying with the idea maybe that was happening, and I had kind of dismissed it because I thought, well, it's got this extra stuff on its nitrogen atom. It probably won't work. Was my immediate thought. And I think that's what the chemists are thinking too. They're saying, no, it can't happen. Forget that. And they won't think any further. But once Anthony kind of teased me and said, yeah, you should take a look. And then I started looking hard. And I can remember that December, I was here in Kauai where I am now, and I just poured over all these glycine papers and finding glycine mutations that were causing diseases that are going up exactly in step with glyphosate. I started to think, holy cow, this really works. All the pieces really came together very quickly over that period, very fruitful period of a couple of months. And we wrote a paper together. That was the first paper that I wrote on the topic of the idea that glyphosate might be substituting for glycine during protein synthesis. So let me tell you a little bit more about the biology of it. It's quite interesting. Glyphosate is an amino acid, and there are about 20 amino acids that are the building blocks of proteins. They're super, super important in the body because the proteins are the workhorses. They're the enzymes, the transporters, the ion channels, the receptors, they're structural proteins. I mean, they really are almost the most important thing in living species is the proteins, which are assembled according to the DNA code. So the genetics is what's dictating how your proteins look. And the proteins have about 20 different amino acids in them. They're lined up like beads on a string according to the code. And one of those 20 is glycine. Glycine is the smallest amino acid, no side chains. And then the way that it detects that it's glycine is because it fits very snugly into this pocket, and all the other amino acids are too big. And it turns out that glyphosate is also the same, no side chains. So it also fits in that pocket perfectly because it is a complete glycine molecule. The difference is that it has extra material stuck on its nitrogen atom, which is outside of the pocket, because the nitrogen atom has to hook up with the paper dolls. They're all joining hands. The nitrogen is outside of the socket, ready to hook up. And the glyphosate fits beautifully in there. And as long as there's room for the extra material, it can go in. And in certain situations, I think it's going to be attracted to the extent that it could be more likely to go in than glycine would at the same place, just because of the particular situation in the context of that particular protein. So I talked about that in the book. It's really quite fascinating because I called it sort of a glyphosate susceptibility motif that I defined.
Mm-hmm. Yeah.
And then I could identify which proteins match that motif. And then you find the protein, you say, well, does this link to one of the diseases? Oh, sure, it does. Here's your fatty liver disease. So it's quite fun too. I'm still playing that game because it's a huge space. And I still find new proteins that are potentially affected by glyphosate and that then link to diseases. I haven't finished that yet. It's just such a huge task. And then, hard to remember all of them, but I certainly singled out a number of them in the book.
Yeah.
And it becomes very clear to me when you see what glyphosate is doing, and you can observe experimentally which proteins it suppresses. They know there's a number of different proteins that get suppressed by glyphosate, and they fit very well with my motif. So it's quite exciting that way. The whole puzzle comes together very nicely.
Yeah, I mean, you've talked about the paper that Anthony Samsel found from Monsanto with the bluegill sunfish.
Yes,
yes. How everyone doesn't accept that it's substituting for glycine after that, I don't know.
I know.
But can you talk through that study a little bit?
That was quite amazing because it was bluegill sunfish, and they were curious to find out whether glyphosate was accumulating in the tissue. It's a very important question to answer. They're doing all these studies on it. This is early data, like 1980s, part of the process of determining if glyphosate is safe. And they fed these bluegill sunfish, they exposed them to radio-labeled glyphosate, which allowed them to trace the radio label independently of the glyphosate. And then they looked in the tissues, and sure enough, there was the radio label in the tissues. Pretty much all the tissue samples they looked at had radio label. And then they said, well, let's just do the standard measure for glyphosate and see how much is there according to that. And it was only about 20% of the radio label that could be accounted for as glyphosate. So now the label's there, but it's not glyphosate. So what is it? And they got the brilliant idea of digesting the protein. So they basically added enzymes that break proteins down into individual amino acids. And after they did that, they were able to increased the yield to 70%. 70% of the radio label could be explained as glyphosate once they broke down the proteins into individual amino acids. And they wrote, perhaps the glyphosate was being incorporated into the protein. This is their words, which is exactly what we're saying. So I feel like they knew. I feel like maybe the people at Bayer, at Monsanto, know that this is happening even today.